In the 20th century, an English mathematician, Godfrey Hardy, and a German physician, Wilhelm Weinberg, founded the Hardy Weinberg Principle, also known as Hardy Weinberg equilibrium. Using mathematical modeling based on probability, they stated that allele and genotype frequencies in a population remain in genetic equilibrium from generation to generation unless specific disturbing influences are introduced. In other words, the totality of alleles of all genes in a population is called the gene pool, which remains constant for a given population. The conditions assumed for the Hardy-Weinberg principle are a very large population, no mutations, random mating, and non-occurrence of migration. Hardy and Weinberg developed a simple equation known as the Hardy-Weinberg equation to study the genetic equilibrium of a given population. Let's understand how this equation is derived. The sum total of all the allelic frequencies is 1. Generally, there are two forms of a gene. That is, two alleles for one character. Each of these alleles has an equal chance of getting inherited. Therefore, the frequency of the alleles would be 0.5 plus 0.5, which is equal to 1. Let us name some individual frequencies as P and Q. In a diploid individual, P represents the frequency of the dominant allele capital A, while Q represents the frequency of the recessive allele small a. Therefore, we can also say that in a population, the frequency of a homozygous dominant individual, that is A capital, is simply P square. In other words, the probability of allele capital A with a frequency of P to appear on both the chromosomes of a diploid individual is defined as the product of the probabilities, which is P into P or P square. Similarly, the frequency of a homozygous recessive individual or small a is Q square. Since there are two ways of forming heterozygotes, or capital A small a, that is, capital A allele from the mother and small a allele from the father, and vice versa. Therefore, the frequency of a heterozygous individual or capital A small a is 2pq. The sum of all these frequencies is dominant plus recessive plus 2 heterozygous dominant, which can also be expressed as capital A plus small a plus 2 times capital A small a. On substituting capital A with P and small a with Q, we get P square plus Q square plus 2pq. Therefore, we get the equation P square plus 2pq plus Q square is equal to 1, which is in fact the binomial expansion of whole square of P plus Q. When the frequency measured differs from expected values, the difference or the direction indicates the extent of evolutionary change in a population. For example, generally, the frequency of P or Q is 0.5. If it changes to either less than or more than 0.5, it would indicate evolutionary change. We can learn about the occurrence of evolutionary change, the direction of evolution, and the evolution rate of the selected trait by comparing the genotype frequencies of the next generation with those of the current generation. Genetic equilibrium serves as a baseline against which we can measure genetic change. However, it is affected by several factors such as gene migration or gene flow, genetic drift, mutation, genetic recombination, and natural selection. Migration impacts the equilibrium because when a population moves from one place to another, there are changes in the gene frequencies of the old and the new populations. Therefore, while new genes are added to the new populations, they are lost in the old population. When this gene migration takes place many times, it is called gene flow. Another factor that affects genetic equilibrium is a genetic drift, which is a change in the relative frequency with which a gene variant occurs in a population due to random sampling and chance. Mutation is another factor that affects the genetic equilibrium. On some occasions, the change in allele frequency due to mutation is so different in the new sample of the population that they become a different species. In this case, the original drifted population becomes the founder, and the effect is known as the founder effect. Genetic recombination leads to variations in offspring, and if this variation is naturally better selected than the parental genes, it leads to speciation over a period of time. Natural selection also affects genetic equilibrium. It is a process in which future generations inherit traits with variations from their ancestors. For example, Darwin's finches of the Galapagos Islands. These heritable variations help organisms adapt themselves to the current environment, increase their chances of survival, and reproduce more successfully than others. Critical analysis shows that variation due to mutation or recombination during gametogenesis, or variation due to gene flow or genetic drift, results in changed frequency of genes and alleles in future generations. In fact, natural selection makes the existing population look like a different population. It can also give rise to situations such as stabilization, disruption, and directional change. Let's look at the distribution curves, which help us study the evolutionary process of the existing population. These curves also show how natural selection operates in various situations that impact a population. In the given diagram, the black dots represent the people who have died before passing on their genes. Notice that in stabilization, the extreme ends of the distribution are removed and everything moves closer to the middle. In other words, more individuals acquire the mean character value, which represents the highest number of the population with a particular trait. Natural selection can also lead to directional change. In the curves, Notice that fewer people are able to attain the mean character value for a particular gene due to depopulation. Natural selection can also lead to a disruption situation, where a greater number of individuals acquire peripheral character values at both ends of the distribution curve. This situation occurs when the individuals at both ends of the curve have a higher survival probability than those in the middle. Evolution is a slow and sophisticated process. The Hardy Weinberg Principle helps understand evolution, as it provides a baseline to measure the genetic change in a population.